Hello there, my name is Valeria Espinosa Salivar from Team 3 in the bacteriology course, and today I will present the technique for detecting Corynebacterium diphtheriae using an immunochromatography strip test. Corynebacterium diphtheriae is a gram-positive, non-motile, non-spore-forming bacillus. Under the microscope, it appears as slender rods with club-shaped ends, often arranged in V or palisade formation, also known as Chinese letters due to their characteristic angular grouping. According to the Libel and Clark model, diphtheria follows the typical pattern of infectious diseases. The agent Corynebacterium diphtheriae, especially toxigenic strains. Diphtheria is caused by Corynebacterium diphtheriae and it's primarily spread through respiratory droplets when an infected person coughs or sneezes, but the transmission can also occur through direct contact with infected skin lesions. Susceptible individuals are those unvaccinated or uncompleted vaccinated with the DTaP vaccine, especially children. Risk factors include overcrowding and poor sanitation, but it increases good immunodeficiency. Diphtheria thrives in temperate and colder climates, and the bacteria also can survive on surfaces. Initially, bacteria colonize nasopharynx and start producing the dangerous diphtheria toxin, and the immunochromatography can potentially detect the bacteria even at this early stage before the obvious symptoms appear. The clinical stage is marked by pharyngitis, fever, general malaise, and most notably, the formation of a pseudomembrane in the throat. Diphtheria can lead to severe complications like myocarditis, which is inflammation of the heart muscle, and neuritis, which is inflammation of the nerves. The diphtheria toxin can cause lasting damage, leading to a permanent cardiac or neurological issues. Unfortunately, diphtheria can be fatal due to the airway obstruction from the pseudomembrane, heart failure from myocarditis, and neurological problems. With prompt treatment, patients can recover. The main virulence factor of Corynebacterium diphtheriae is the diphtheria toxin, encoded by the tox gene, which is carried by a lysogenic bacteriophage. The toxin is an Ab exotoxin that inhibits protein synthesis in host cell by inactivating elongation factor II, leading to the cell death. Other virulence factors include adhesins that help the bacterium attach to the epithelial cells and mechanisms to resist immune responses. The target molecule detected by the strip-based immunochromatography test is the diphtheria toxin, produced by the toxigenic strains of Corynebacterium diphtheriae. This diphtheria toxin is a protein-based exotoxin that has a binary Ab structure. The B subunit is responsible for recognizing and binding to the epidermal growth factor receptors on the surfaces of the human cells, and this facilitates the internalization of the toxin. The A subunit is the enzymatic component. Once inside the cell, it inhibits protein synthesis by inactivating elongation factor II, a key factor in translocation. This inhibition results in cell death. Biochemically, the diphtheria toxin is head-labeled, highly antigenic, and also is capable of exerting cytotoxic effects at very low concentrations. These properties make it suitable target for immunological detection methods, as it can be specifically recognized by the antitoxin antibodies used in the diagnostic assay. The diphtheria toxin plays a central role in the pathogenesis but not in the survival or replication of Corynebacterium diphtheriae itself. In other words, the toxin is not essential for the bacterium's growth, colonization, or life cycle in basic sense, but it is crucial for the disease manifestation and transmission. Now let's explain the mechanism of the test. The immunochromatography strip test works by capillary action. First, the sample is applied to the sample pad and follows toward the conjugate path, where it meets antibodies labeled with colored particles. If the target analyte is present, it binds to these labeled antibodies, forming a complex. This complex continues migrating to the test line, where immobilized capture antibodies trap it, producing a visible colored line. The fluid keeps moving to the control line, where excess labeled antibodies are captured, confirming that the test worked properly. A positive result shows two lines. A negative result shows only the control line, and if no control line appears, the test is invalid. The matrix of the immunochromatography strip test is usually made of nitrocellulose membrane, which allows capillary flow and immobilization of antibodies. Other support materials include the sample pad made of cellulose or polyester, the conjugate pad made of fibrous glass or polyester, and the absorbent pad made of cellulose. The principle of the test is based on lateral flow, where the sample migrates through the strip by capillary action, binding to the labeled antibodies. If the target analyte is present, it forms a complex that is captured at the test line, producing a visible signal. The test begins with the sample, which is usually the supernatant from a culture of Corynebacterium diphtheriae. Sometimes this sample is first mixed with a buffer solution like Tris HCl or phosphate buffer to maintain a stable pH. As the sample travels, it dissolves in lyophilized monoclonal antibodies located in the conjugate pad. They are specific to the diphtheria toxin and are conjugated to gold nanoparticles that serve as visible markers. If the toxin is present in the sample, it forms a toxin-antibody gold complex. This immune complex continues migrating toward the test line, which contains immobilized secondary antibodies that also recognize diphtheria toxin but bind to a different epitope. As the liquid continues to migrate, it reaches the control line, which contains anti-IgG antibodies. These antibodies bind to the gold-labeled antibodies. The test should be done at room temperature between 15 and 30 degrees Celsius. After applying the sample, you have to wait about 15 to 20 minutes before reading the results. The sample used is typically a throat swab, nasal swab, or skin lesion swab. Sample must be collected using a sterile swab and transported immediately to the laboratory in special transport media, such as Amis medium or Loeffler's medium to keep the bacteria alive. If immediate processing is not possible, samples should be stored at 4 degrees Celsius and processed within 24 hours. If the sample is positive, you will see two color lines, one at the test line and one at the control line. If the sample is negative, you will only see one line at the control line, and if the test is doubtful, usually it's because the control line doesn't appear. This immunochromatography test has a high sensitivity, usually around 90% or higher, and a high specificity, typical also above 90%. The main advantage of the immunochromatography test are that it's very fast, giving the results in about 15 to 20 minutes, is easy to use, is portable, plus it's safe and also is cost effective compared to more complex tests like PCR. While this test is quick and easy, it does have some disadvantages. For one, it can have cross reactions with other bacteria or pathogens, which might lead to the false positives. Another downside is that the sample handling can affect the results. Also, there may be issues with residues, so proper disposal and cleaning are important. Currently, immunochromatography strip tests are not widely available commercially. However, some companies offer ELISA kits for detecting diphtheria toxin antibodies. For example, Abcam sells indirect ELISA kit for human diphtheria toxin IgG, or IBL American sells quantitative IgG ELISA for diphtheria toxin. Prices vary depending on the supplier, but may range from $400 to $800. That's all for today. I hope this video gives you a better understanding for the immunochromatography strip test for diphtheria. Thanks for watching.